In today's session, let us discuss about the next topic in biomolecules, that is 9.6 nucleic acids, and we'll complete 9.7 also, which is proteins. Let's start our class. So, the next topic is 9.6 nucleic acids. Now, in the acid insoluble fraction, another type of biomacromolecule we find is nucleic acid. Now this nucleic acid, what is it then? It is a biomacromolecule. Means its molecular weight is more than 10,000 daltons. And next point, question what we have to ask is, it is a homopolymer or heteropolymer? Nucleic acids are heteropolymers. They are heteropolymers of nucleotides. A chain of nucleic acid is called as a polynucleotide. When we take a polynucleotide chain and when we try to break it, then what do we get? Means, if you take an enzyme called nucleotidase, which breaks a nucleotide, then we get a nucleoside and a phosphoric acid, phosphate. So, when we break a nucleotide, we get nucleoside and phosphate. When you break a nucleoside, then we get a sugar with nitrogen bases. So, sugars are of two types which are found in nucleic acids. Of course, the sugar is pentose. It is aldopentose, aldehyde group sugar with 5 carbons. Now, if it has all the hydrogens, then it is called as ribose. Hydrogens and oxygens. If you remove one oxygen, then that too from the second place, then you call it as 2-deoxyribose. If the sugar is ribose, then such a type of nucleic acid is called ribonucleic acid. If the sugar is deoxyribose, then that nucleic acid is called deoxyribonucleic acid. It is ribonucleic acid, and this sugar is deoxyribonucleic acid, DNA full form, RNA full form, right? So sugars are of two types which are found in nucleic acids. They are riboses and deoxyriboses. Nitrogen bases are also two types. They are purines and pyrimidines. Nitrogen bases are of two types called purines and pyrimidines. Now, purines are of two types called adenine and guanine. And pyrimidines are of three types called thymine, cytosine, and I wrote it on the top, uracil. So, adenine and guanine are examples of purines. Thymine, cytosine, uracil are examples of pyrimidines. So, what is adenine? What is the chemical name of adenine? Adenine is called as 6-aminopurine. Guanine is called as 2-amino-6-oxypurine. It is 2-amino-6-oxypurine. Now, in a purine ring, if you substitute amino group at the 6th place, then it becomes a nitrogen base called adenine. If you substitute amino group at the 2nd place followed by oxy group in the 6th place, then it becomes guanine. Means to tell that adenine and guanine are substituted purines. So let us write down that adenine and guanine are substituted purines. Thymine, what's the chemical name? It is 2,4-dioxy-5-methylpyrimidine. So thymine is 2,4-dioxy-5 methyl pyrimidine. And what is cytosine? Cytosine is 2-oxy-4-amino-pyrimidine. So cytosine is 2-oxy-4-amino-pyrimidine. What is uracil? Uracil is 2,4-dioxypyrimidine. I'll write on the top. It is 2,4-dioxypyrimidine. Means for the pyrimidine ring, if we are substituting methyl groups and oxy groups and amino groups, then it becomes thymine, cytosine and uracil. Can we then say thymine, cytosine, uracil are substituted pyrimidines then? Yes. So as we told, adenine and guanine are substituted purines. Likewise, thymine, cytosine, uracil are substituted pyrimidines. They are substituted pyrimidines. Now, we have seen sugars, two types of sugars, either ribose or deoxyribose. We have seen bases, two types of bases, purines and pyrimidines. Purines of two types, adenine, guanine. Pyrimidines, three types, thymine, cytosine, uracil. Now, sugar is joined to a base by glycosidic bond. Sugar, if you join to base, by which bond? Let me write it here. By glycosidic bond. You will get a nucleoside. Nucleoside joins to phosphate by an ester bond. You will get a nucleotide. One nucleotide joined to another nucleotide, another nucleotide, another nucleotide to make a polynucleotide by what are these bonds? They are called phosphodiester bonds. So this is our basic introduction. So now if you see, they tell in NCERT, it tells like nucleic acid is a polynucleotide. Polynucleotide has three components. One component is the sugar. The second component is the base. The third component is the phosphoric acid. So if one phosphate is attached, monophosphate it will be formed. Two phosphates, diphosphate it will be formed. Three phosphates, triphosphates it will be formed. So if we have to see the structure, in the next topic we can see the structure when we talk about nature of linkage of biopolymers. In that linkages we will see. Now we can get to the next topic 9.7, which talks about proteins and the structures of proteins. Let's talk about the next topic, proteins. Now proteins, if we have to discuss, they are also biomacromolecules like polysaccharides, polynucleotides, right? Polypeptide means protein. Polypeptides are also, so they are also polymers, true polymers. You will write down as true polymers. They are also true polymers. Okay. Proteins, if we see, now if you look in the point of inorganic chemist point of view, for an inorganic chemist, the structure refers to writing a molecular formula. So the structure means, it means writing a molecular formula. It means writing a molecular formula. Molecular formula like NaCl, MgCl2, like that. Whereas if you think in terms of organic chemist, now organic chemists, looks at the two-dimensional view of the structure of a compound in the sense like a benzene ring or heterocyclic ring like that. An organic chemist looks at two-dimensional view of a structure. Let it be a benzene structure or a naphthalene structure, a two-dimensional view it is. Now, if you take in terms of a physicist, they view the three-dimensional angle of a structure. At three-dimensional view. Whereas biologists look at the four-dimensional view of a structure, look at primary, secondary, tertiary and quaternary structures of proteins. So when it comes to biologists, look at four-dimensional view of structures. Now, when we have to talk about the four-dimensional view of structures of proteins, now let us go. So, we have four structures called primary structure, secondary structure, tertiary structure and quaternary structure. So, primary structure is nothing but the linear chain structure. So, when we have to talk about the primary structure of a protein, it is nothing but a linear chain of amino acids. You know, they are joined by peptide bonds. They are joined by peptide bonds. For our understanding, we will see they have given in the NCERT. So they went with figure 9.3. If you look at the figure 9.3, they have given us the structure, primary, secondary, tertiary, quaternary structures. There, they've shown us the diagram in this manner. Let us try to draw it. The amino group, alpha carbon, R group hydrogen, and the carboxylic group, and this is the carboxylic group, and here again this is an alpha carbon, hydrogen, then again this is a amino group, again alpha carbon, and again C double bond O, NH, right, C double bond O, then we have to give alpha carbon and then NH2. In this manner they tried us to show the primary structure, which they are telling it's a linear structure. Now see, I drawn glycine. Till here if you see, this is one glycine molecule. And another glycine molecule. Till here it is one glycine molecule. C double bond O reacts with NH. C double bond O if you are giving here, then we need to go with NH here. And then we need to go with alpha carbon. Then C double bond O. Then C double bond O, if it is there, then this is NH. Then this is alpha carbon. And this will be C double bond O, OH. So what I draw here is one glycine. And this is alpha carbon. Another glycine I am showing here. And this is the third glycine molecules. Means I draw a tripeptide here with three glycine molecules. So this is one glycine, second glycine, a third glycine molecule. Just to show you. Now, how did we make a bond? A glycine has an amino group. It has a carboxyl group and its R group is hydrogen. Its R group is hydrogen. So I think we need to show another hydrogen because it's H2. Yeah. Now if you see, this is a linear structure. Now this is a left hand end. And this goes right hand end. This goes right hand end. Now left hand end, if you see, which functional group is free here? All the hydrogens are free. Amino group is free here. its carboxyl group is involved in a peptide bond linkage. Right hand side, if you see, which group is free here? C double bond O, OH group is free here. That is why for a protein, in a linear chain, a protein molecule, if you say the left hand end, you can also call it as an N-terminal end. Or you can call it as amino group. It is also called amino terminal end. And this end where the carboxylic group is free is called as C terminal end. Or we can call it as carboxylic terminal end. N-terminal end is the place where the first amino acid will be found. C-terminal end is the place where the last amino acid is found. What is this? This is the line diagram of a protein. Which conformation does it represent? It represents the primary conformation. Primary conformation means linear conformation, where the adjacent amino acids are joined by peptide bond. So, in the next topic, linkages, we will see how to make a peptide bond. When we make a peptide bond, the first amino acids, amino group will be left free. It is reactive with acids or bases. So, that's why, since the amino group is not disturbed, it is free, we call it an N-terminal end. This is a primary structure. In primary structure, what is the bond we find here is peptide bond. The only one bond which is seen in primary structure is peptide bond. Example for primary structure is insulin molecule. Hormone, what is that? Pancreatic hormone made up of two polypeptide chains, A chain and B chain. A chain is made up of 21 amino acids and B chain is made up of 30 amino acids. So this is called as A peptide and this is called as B peptide. Now, this A peptide looks like one primary structure and the B peptide also looks like one primary structure. So, we can take insulin as an example for primary structure of a protein. Now, let's go to the secondary structure of the protein. Now, when we have discussed about the scientist information, then we talked about Linus Pauling, who worked about the alpha helical and beta pleated sheets, and Ramachandran got influenced and he proposed the collagen triple helical structure. So, right? So, when we are talking about secondary structures, who explained the secondary structures of proteins means Linus Pauling. What are the two types of secondary structures means? They are alpha helix and beta pleated sheets. Now, let's go to the secondary structure. So, we have seen primary structure and we have taken the example of insulin there. Insulin's individual polypeptides we have taken. Now, secondary structure which was explained by Linus Pauling, he told there are two secondary structures. In figure number 9.4, they have given the picture. In figure number 9.3 also they have given the picture. In figure number 9.4 they have shown us the secondary alpha helical structure in this manner. And then they are telling in proteins which type of turns, right-handed helix or left-handed helix. So there are only right-handed helices found in proteins. So protein chains show right-handed helices only. They show right-handed helices only. So we discussed they are L-alpha amino acids and they are right-handed helices. So this is the alpha helical structure then. This is the alpha helical structure of a polypeptide or protein molecule which is turned in a right-handed direction, okay, which was explained by Linus Pauling. Now, one helix below the helix, below that one more helix, below that one more helix, below that one more helix. So, if they should be kept in proper manner like this means, apart from peptide bond, you need an additional bond. That additional bond will be hydrogen bond. Now, since a helix, per helix, you know, approximately 4 amino acids will be there. approximately 4 amino acids here, 4 amino acids here. Now for our understanding, first amino acid, second amino acid, third amino acid, fourth amino acid, fifth amino acid, sixth amino acid, seventh amino acid and eighth amino acid, like that they are. Then if you have to go 9, 10, 11, 12, like that. First amino acid is linked to fifth by a hydrogen bond. Second amino acid is linked to sixth. 4 is linked to 8. 3 is linked to 7. So these bonds which I am drawing, which will make them to keep one below the other, is called as hydrogen bonds. This is intra-hydrogen bonds. So what is that additional bond which stabilizes a little compact structure? It is hydrogen bond. Now if the bond is between the chain, within the chain, it is intra-hydrogen bond. It's intra-hydrogen bond. In addition to peptide bond, hydrogen bonds are also seen in secondary structure. Now when we want to talk about the second structure which Linus Pauling only explained, which is called as beta pleated sheets. Beta pleated sheets. So to explain a beta pleated sheet, take a polypeptide. I told you N-terminal and C-terminal are the ends of a polypeptide chain. If all the polypeptides are having N-terminals towards one side and C-terminals towards one side, then that is called as, so all N towards one side, all C's towards one side. So one amino acid joined by peptide bond and they are in turn connected by hydrogen bonds. They are in turn connected by hydrogen bonds. Such a type of beta pleated sheath is called as parallel beta pleated sheath. Sheath, secondary structure. And if like one chain goes this way, the other chain goes this way, I am doing anti-parallel. N-terminal, C-terminal, N-terminal, C-terminal, N-terminal, C-terminal. Adjacent amino acids by peptide bonds and hydrogen bonds. Now this is called as anti-parallel beta pleated sheets because N and N are not there. N is with C-terminal end. So I'll call it as anti-parallel beta pleated sheets. Now this is one type of secondary structure which is called as alpha helical structure, which are right-handed helices, and this is second type of structure which is called as beta pleated sheets. Okay. Now this hydrogen bond is called intra-hydrogen bonds because the bonds are within the chain. These hydrogen bonds are between the chains, so we can call these hydrogen bonds as inter-hydrogen bond. So primary structure only peptide bond, secondary structure peptide bond and hydrogen bond. Now coming examples for secondary structure are keratin protein which is present in the hair. The examples are keratin of the hair, the myosin of the muscle, collagen also, which was explained by Ramchandran, is also an example of secondary structure. That also I am writing down. Collagen in animal tissue are examples of secondary structures. Now let's go to the tertiary structure. The tertiary structure is very important. The tertiary structure is the stage where amino acids are, the proteins are active. So we call it as active conformation of a protein. It is active conformation. It is the three-dimensional structure. Tertiary structure is called three-dimensional structure. It's also called the native conformation of protein. It's called native conformation of the protein molecule. The enzymes are too much active in their tertiary states. Okay. Now tertiary structure, if we have to see, the example goes is myoglobin, made up of only one polypeptide chain. Made up of only one polypeptide chain. So a linear chain, we are calling it as primary structure. A linear chain coiled secondary structure. Now further coiling like a hollow woolen ball-like structure. So I am just taking it and we are further coiling it. We are further coiling it. Like a, I try to draw a hollow woolen ball-like structure. Like a hollow woolen ball-like structure becomes the tertiary structure. To make a linear chain, peptide bond enough. To make a helical chain, peptide bond with hydrogen bonds are required. To make a tertiary structure, a hollow woolen three-dimensional ball-like structure along with peptide bond, hydrogen bond, we need van der Waals forces, hydrophobic attractions or interactions, disulfide bonds. All such bonds are required to convert the structure into a globular structure, which is called tertiary structure, which is called globular structure. And the example goes is myoglobin. Now coming to the last structure, which is quaternary structure. The last structure, I will keep it here, is called quaternary structure. A beautiful example for quaternary structure is hemoglobin, made up of 4 polypeptide chains. 2 chains are of two alpha chains are there, and 2 beta chains are there. Alpha chains are made up of 141 amino acids, beta chains are made up of 146 amino acids. Each chain is a tertiary structure like this. Now we need to draw them. Just trying to make it a 3-dimensional view so that you can understand. So this is one woolen ball. Let me draw another woolen ball. This is one woolen ball, another woolen ball, another woolen ball, another woolen ball. So here I am trying to show 4. This one is one alpha chain. Here is beta chain. Here is beta chain. Here is alpha chain. And this is one tertiary, another tertiary, another tertiary, another tertiary, interconnected by hydrogen bonds, hydrophobic bonds, disulfide bonds with each other. Then it makes a quaternary structure. So quaternary structure, example for quaternary structure is hemoglobin, which is made up of 2 alpha chains and 2 beta chains. So this is the structure. 2 alpha chains will be there, 2 beta chains will be there. So it goes like alpha between 2 beta, beta between 2 alpha, and these chains are further connected by all these forces. So in this lecture, what we discussed is, we have discussed the structures of nucleic acids and the structures of proteins. Now in the next lecture, we can discuss about the next topic. So if you like the video, you can like, share and subscribe. Thank you.